Thanks, Erica.
Yay!
Yay
Let's
from
all welcome
me.
Andy. Yes. Sorry, I like applauding. I'm going to make people applaud.
Hello. Okay, this is going to be quite different to yesterday. Bit of light relief. We're going to lean into the naturalistic side of intelligence rather than the artificial side of intelligence. I spend my life working with that organism, not just because it's incredibly beautiful, but because it's fascinating for a number of reasons. What's been interesting me for my entire career is this thing does a lot with very little. It has a minute brain by most standards. By a fly brain, it's gigantic, but by any other standard, it's a tiny, tiny brain. And yet it seems to achieve a prodigious amount. And particularly with reference to where we're going now with current robotics approaches, which are leaning more and more and more onto massive compute. And someone like me is constantly going, well, that's fine, but if a bee can do more than our current autonomous robots can do with absolute minimal autonomous compute, isn't there something that we could learn from that? And that's the kind of theme that I want to pick up in the talk today. So the behavioral capacity of a bee is quite phenomenal. And I have sometimes illustrated that with this gigantic weird-ass laundry list of things it's claimed bees can do. It's actually more impressive to think about what they have evolved to do in their everyday life. So a bee excels at a task that I could not perform, which is a bee makes a living foraging on flowers. It's able to make an energetic profit harvesting minute amount of cryptic pollen and nectar, which are these ephemeral cryptic resources dispersed across the environment. The distribution is highly unpredictable. The bee is having to learn where it can find these resources. And it's not just a matter of looking at a meadow like this and going, well, which type of flower is going to give me pollen and nectar? No flower gives pollen and nectar 100% of the time. It's a matter of learning what stages of which types of flower at what times of day under what weather conditions are most likely to offer pollen and nectar, as well as the properties of the pollens that have got to be lined up with the demands of the hive. Less so the properties of the nectar, they're all sugar, but nectar is highly, highly ephemeral. We're talking about millimetric amounts per flower. As well as that gigantic multidimensional classification problem, we have webbed into this a crazy foraging economics problem of at what point have I decided I've exhausted this patch and should I move to a different patch where I can't predict the state of that other patch? There's that element too. And then also tied into that, there's also an energetic navigational problem in that the bee is flying potentially 6 or 7 kilometers on this foraging circuit, and it's got to return with those resources to a minute fixed point in space, the hive entrance, which is itself also cryptic. It's got to do all of that while making a profit, and if it gets it wrong, it will die. So a bee only has enough onboard resource to sustain 15 minutes of flight. If it can't refuel in flight, it will die. Hello, yes, question. Good.
Is your word cryptic a key jargon word, or is it just meaning hard to understand? Or what is the word?
Cryptic meaning none of these flowers really sort of wave their pollen and nectar in the air. The bee has to sort of learn how to access and search within each flower to find them. And flowers do that because they like to cheat. They want a bee to visit without always giving a resource. So don't advertise if you've got pollen and nectar. Make the bee hunt for it. That's sensu cryptic. So the bee is doing all of that with that as its only onboard processor. That's a brain with just shy of 1 million neurons, 960,000 neurons. It's millimetrically scaled. It's small, but it's not simple. It has this fantastic modular structure. That's a micro-CT scan. It's emphasizing the regions that in insect neuroscience we call neuropil, which are the regions of connection and computation in the insect brain. So this is where the synapses occur. The space between is filled up with the cell bodies of the neurons. So highly modular brain, minute structure, just under 1 million neurons. The reason I like a brain like this is compared to a mammalian brain, it is highly comprehensible. We have now a very good idea about what each of these modules do and the internal neural circuitry of each of these modules. And yes, mate, there's a question there.
Can you point out the olfactory and the visual parts of that
See,
brain, or is
that's
it
why I
hard?
like an insect brain, because I can, in
I was wondering
absolute
if you
terms.
could, yeah.
So olfactory processing here, antennal lobes, primary olfactory processing. We have 3 lobes for visual processing. Here, 1, 2, 3, 1, 2, 3. And then these sensory modules each project into the central brain where we have the multimodal integration centers. What's amazing about the insect brain, and it's fantastic for doing any sort of computational modeling of the insect brain, there are only 2 multimodal integration centers in the insect brain. The mushroom bodies that were mentioned yesterday by Sanjoy, and then the central complex, which I'll talk about a little bit more here. The mushroom bodies are here, multimodal integration centers, so we get visual input, no, we don't, yes, visual inputs here, olfactory inputs here, and then the output is to pre-motor regions down here. The honeybee invests about 1/4 of its neurons in those mushroom bodies, so about 250,000 neurons into the mushroom bodies, so very much bigger than the fly mushroom body, but the same sort of architecture that Sanjoy introduced yesterday. We're basically expanding sensory information into a high-dimensional space, which is very, very good for classification problems. What's also nice about the insect brain, very intensively studied, nowadays relatively very well understood. I think certainly per neuron better understood than any mammalian brain, which is useful. That's the mushroom body here is a micro-CT scan. These are light micrograph drawings of the mushroom body by Félix Dujardin from 1860. So some of the very first light micrographs were of the honeybee brain, and the accuracy with which he was able to draw the mushroom body sitting at the top of the brain, at the back of the brain, is, I think, quite phenomenal in terms of how accurate he got it. So we've had, over 150 years of neuroanatomical study and neurobiology of the honeybee to work out what these things are doing. It's useful and it's appropriate and it's accurate to abstract the computational architecture of the mushroom body as a simple perceptron. Sandra introduced this fantastically yesterday. We have sensory inputs. This is a model that James Cope and Alex Marshall developed for the mushroom body as well. We have a sensory input population of the input of the mushroom body, the high-dimensional space that expands that information over the Kenyan cell population, which in the case of the honeybee is about 250,000 neurons, and then that compresses to a very, very small number of premotor output neurons. Here we just caricatured them as go, no, go. In biology, though, it collapses down to just a few hundred neurons at the output from the honeybee mushroom body, so a massive convergence of the mushroom body output. This type of structure is, that perceptron structure was used in early machine learning models as being extremely good at classification problems. We think it does the same kind of function for a bee. And I'm going to take a little bit of a segue here into just some, as Erica said, it's the chillest day imaginable today. So we're just going to drift off into some impressionist art just to show how good bees are at solving classification problems. This was a toy experiment, not an experiment, it was a toy demonstration that I did for a documentary in Australia, just to see if I could train bees to classify paintings by artists, to recognize different styles of impressionist art was how we sold it. So I took 3 impressionist painters, Emily Carr, what am I doing? I'm breaking the pointer, good. Emily Carr, Claude Monet, Georges Seurat. Now, each has a clearly distinctive style to our eyes and to the way that we process information, but they're all painting in tones of blues and greens. They've all got blue skies and green things at the bottom. They've all got vertical naturalistic elements in the paintings. The question was, could we train a bee to distinguish painting by artistic style? And the way you do that with a bee is the first thing you do is you stick a number on the bee so you can tell who's who, and then you train the bee to come into a small arena and to pay attention to little tiny yellow squares. You do that by putting sugar water on the yellow squares, and you get your bee coming into the arena and hunting on yellow squares for sugar water. And then once you've done that, we set up in the arena a matching to sample task. So we had a painting by one of the artists at the entrance to the arena, and then within the arena, an array of paintings by all 3 artists. And let's say that we put a Monet at the entrance. In the arena, on different paintings by Monet, we put sugar, and on all the other paintings by the other artists, we put quinine. Now, quinine tastes bitter to a bee, it's not harmful, but what it means is that if she sees Monet at the entrance and lands on a different Monet painting, she gets a sugar reward. But if she lands on any other painting, she just gets a bitter taste and will fly off and keep hunting. So we're trying to get the bee to learn a rule to form a category of paintings according to the painter. This is what one of those training trials looks like. This was a Monet training trial. So you see a Monet at the entrance. There's quinine on the non-Monet paintings, and then sugar water on the Monets. And you get these beautiful, beautiful searching flight patterns, which I really do want to analyze in detail at some point. That's an Emily Carr. She lands on that tastes bitter, so she just rejects and flies off, keeps flying around, wobbly camera, and then lands on the Monet. Now she can feed. She'll feed to repletion. She's released from the arena. That's one training trial. Now, we only gave the bees 18 training trials with this structure, just 18. And then we moved to the test phase. In the test phase, painting by one of the artists at the entrance, array of paintings in the arena, sugar on all of the paintings now, just to make sure they can't be telling a difference remotely between sugar and quinine. And now we're looking at which painting the bee lands on first. So here's a test now. That's an Emily Carr painting. She inspects it. Into the arena she goes, eventually, thank you. And then here we go now in the test. You can see she's inspecting without landing in the test, inspects all the paintings, keeps flying around, and then lands on an Emily Carr and feeds. Ta-da, sample of one, let's publish, let's stop. Thank you, no, don't thank me, thank the bees, I was just an observer. But of course, it is possible here that what we've got is we've learned stimulus-dependent rules, so we thought let's up the ante. With no additional training, we gave the bees 3 paintings the bee had not seen before. So a Carr, a Monet, a Seurat the bee has not seen before. No more training. We're asking now, can the bee classify a novel painting according to artistic style? So here we have an Emily Carr the bee's never seen before. She lands on it, which is a bit weird. So clearly that's threw her off a little bit. Into the arena she goes, sugar on all the paintings. We're looking at where she lands first, and she lands on an Emily Carr. So again, N of 1, hooray, sample sizes and bees, fantastic. Yes?
So these, she did see all these before, she just didn't see the reference?
Yes.
Yeah, okay.
She saw all these before, but she didn't see the reference before. So the question is, can she match a novel reference to the same artistic style? And with an N of 1, we can conclusively conclude that yes, she can. Now, I didn't do this as a full scientific study. Judith Reinhardt, this is purely a demonstration. Judith Reinhardt has done this as a full scientific study, and it does actually hold up, that bees can learn to classify Impressionist artist paintings by style. There's all sorts of ways by which this is possible, because of course these artists differ in their spatial frequency, they differ in the average tones and luminance that they'll be using in their paintings. But what I want to highlight is a bee is able to learn this complex multi-part classification in just 18 learning trials. And I want to come back to this learning speed a little bit later in the talk. Now, with James Marshall, who's part of the program here, we actually wanted to push how well we could understand the classification capacities of a honeybee if all a bee has to learn classification is that simple mushroom body computational architecture. And one of the studies that really, really stood out for me, and one of the ones that inspired me to start working with bees over 20 years ago, was this work by Martin Gierfer that showed that even a honeybee was capable of abstract concept learning. Abstract concept learning seemed to me utterly remarkable. It's abstract. It's abstracted from any of the stimulus properties. So yes, they could solve that artistic task by learning potentially differences in spatial frequency that the artist tended to lean towards. That's embedded within the stimulus structure itself. If it's abstract, it's not part of any of the stimulus structure. And this really bugged me, like how can an organism like a bee possibly learn something that is abstract? Now, the abstract concept that bees were supposed to be able to learn was the concept of same and difference. Same and difference are intuitive to us, but nothing about same is embedded in any of the individual stimuli that a bee might see as it's trained in this task. And yet bees could solve a same and difference learning task. And the way this was done with bees is you used what's called a delayed match to sample paradigm, a delayed match to sample task. So let's say we're training a bee on same. What we do is we have a Y-maze arena that looks like this. We put yellow at the entrance to the arena. If we're training on same, if the bee goes to yellow in the arena, we reward it with sugar. If it goes to blue, which is not the same, we punish it with quinine. And that's one learning trial. Then the bee hops out the arena. We would then put the blue stimulus at the front. If we're training on same, if it chooses blue in the arena, and of course we're counterbalancing the sides and all that kind of stuff to do the good learning stuff that we do. But if there's blue at the entrance and we're training on same, if it chooses blue within the maze, it gets rewarded. If it's on yellow in that context, it gets punished. And you train the bees through those kind of training trials. Yep, David.
I forget if you said it in your presentation of this at DISI a couple of months ago. What happens if it comes in and does the wrong thing, quinine, then immediately realizes that and goes off and then quickly goes to the other one to get the sugar? Do you interfere with it? Do you kill it before it gets there? What do you do?
So yes, first of all, apologies to people who've heard this talk before, just scant months ago. What happened in this paradigm is actually the bee has to fly through these into another chamber that's behind it, not shown on this slide. So its choice would be to fly through this yellow disc, which also has a hole into it, into a small chamber. In that chamber behind, there's little pots, which it can't see or smell until it goes into that chamber that contain either quinine or sugar. And then if it gets it wrong, you release it from there. So it doesn't get a chance to hop back across. Yeah, it's released out. It's got to go back and repeat the maze, which actually is important for the training. So you put bees through this paradigm. You can train them on same and difference in this. You can then test them again. If you're testing, you put empty pots in those chambers behind it. You simply look at what choice the bee makes in a test, and you can show it can learn this same and different paradigm. Yes, there's a question.
I recall something, I'm not a bee expert, but that bees have specializations of different functions in the hive and stuff. Does that impact is that totally behavioral and behaviorally learned, or is there an impact in their ability to do this based on what their role is?
So bees do have a behavioral division of labor, absolutely. The biggest characterization of that is between nurse bees that stay in the hive and don't leave the hive and raise the young, and forager bees that don't raise the young in the hive and spend their life outside the hive foraging. Now, for an experiment like this, all the work is done with the forager bees. And for most of the work that I do, all the work is done with the forager bees. In the case of the honeybee, bees, each individual progresses through those behavioral roles as they age. So it's not genetically determined that there are castes they're born into and that they stay into. They progress through them as they age. There is work that shows that there are differences in brain development between those castes members. There is some work that shows that the nurse bees who are younger are cognitively less proficient at this kind of task than the forager bees. It's as if they are optimizing brain development for the onset of foraging, which is the cognitively most demanding phase of their life. Okay. So to come back to this, it's not enough to say that we trained a bee if it sees yellow to go to yellow and to solve that task, because that could be a stimulus-dependent rule. I see yellow, go to yellow, get reward. I see blue, go to blue, get reward. That's stimulus matching. But what convinced people and convinced me that bees had learned an abstract concept here was that you could train the bees as I described with colors, and you could train them either to match or not match with colors. And then with no additional training, you could swap into the maze, say, for example, these patterns, vertical versus horizontal. And if you trained the bee on same and suddenly you put a vertical grid at the front, a bee trained on same would choose vertical in the maze. So it would match it, even with stimuli it's never seen before. So this transfer to stimuli the bee's never seen before is what convinced people this is an abstract concept. It's not bound to the stimulus, it's just abstracted. You could even train them on odor fields and then swap it into the visual domain, and they could still solve the task, which is really, really quite impressive. So this bugged me for a long time. Yes, mate, there's a question there.
You have any question? Please go ahead. No, I think you raised your hand.
So I'm just wondering, so for these, it was always done with a pair where there was so you'd never have like the vertical lines as the stimulus, and you might have, I don't know, the horizontal the vertical lines or the swirly, or have the vertical, the horizontal lines. But what I'm wondering is, you could never make a mistake where you rotated something. You thought, well, that looks about right.
Yeah. So for this study, and this is actually an important point, for this study, one bee was trained with one pair of stimuli and then testing the transfer to a different pair of stimuli, which contrasts with how you would do this study in macaques. If you were doing it in macaques, you might have 100 reference stimuli you had used in the training for same, and a different batch of like 100 stimuli you're doing different. I think that's actually an important distinction that we'll get to later. There's a question there, mate?
It's maybe a little bit related to that. So how much information can they remember for how long? And let's say if you want to switch from, rewards are associated with same concept, then rewards are associated with different concepts, then can they reset that?
So this is actually quite nice because it ties in with what Sanjay was talking about, catastrophic forgetting. The bee's memory is prodigious and fantastic. So bees can learn a color reward association in one-shot learning, and that can last the entirety of the bee's foraging life. Certainly after 3 exposures, to example, a color and reward or an odor and reward, lasts the entirety of the bee's life. But the bee's life, foraging life, is maximum a week. So what actually limits us in exploring the bee's memory capacity is these foragers don't live long. So we can't train them over hundreds of paradigms. There has been work that's been done training bees on 2 or 3 different rule learning tasks. They can acquire 2 or 3, and they can retain 2 or 3 without them interfering. Yes. I'm going to totally overrun my time, but I'm good. Yep.
There are 2 patterns here. What if there are 3 or 4? If you increase number of possibilities,
Yep.
what happens?
So the question there is, if we increase the number of training stimuli here, what happens? The answer is it hasn't been done. And it actually would be very interesting if we could do it. What we run into is the bee doesn't live long enough for the training. So to get them through this paradigm with just a pair of stimuli, that's 1 full training day. The maximum we can really follow a bee practically is up to 3 days, but students start to die. Students get something called heat stroke. Like I've had to learn, please don't set students paradigms that take 14 hours to run because the students will get heat stroke. So there's practical concerns that we're dealing with, but it's a really good question and it hasn't been done, but it needs to be done for reasons I'll get to shortly. Yes, mate.
Right, the answer is the same, but so this is fascinating. But I'm curious, how much can you push it? Like can you train them to have an indicator for whether they should find the same or different, or like have 2 things in front, and if they're the same, they should go to something that's the same if they're different. I mean, I'm trying to push it see how they can learn.
There's only one study that tried to do that. There's one study that tried to ask, what if we put distractors in front of this? What if we're interfering stimuli in there? And the outcome of that one study is ambiguous. And actually, we're following up on that right now. So we want to ask, and this is maybe answering your question, or it might not be, because you're looking like that's not answering. I'm not answering your question. Why don't you ask it going, right? What's up? Yes.
No, that's also interesting. I'm trying to think of the concept being more complicated rather than the training. Like a concept that in particular cannot represent with a single summation neuron.
Okay. This is one of the most complicated concepts that bees have been tested on. There are other so-called concepts that bees have been tested on, like can they learn above, below? Can they learn greater than, less than? That's actually less complicated than this, because above, below, and greater than, less than can be embedded into the stimulus properties. This would be the most complicated concept. There's a question there, mate?
Yeah. So bees are quite social creatures. Have
Yes.
you looked at this from more of like a social learning perspective and also just incorporated? Like you could kind of maybe test to see how these like same different concept, like kind of how long it lasts in generations maybe?
Yeah. So bees are social creatures. The mechanism for them to communicate this kind of information slowly is extremely, extremely limited. There is one study on whether there's social facilitation of learning, and that used a 2-part mechanical manipulation task. And what the claim in that study was no bee could learn both parts of that, but if bees saw a bee doing one of the movements, that facilitated them learning that, and then because they could learn the other one, they could solve it through social facilitation. And that was thought to be able to perpetuate that. It's been aligned to the idea of cumulative culture, but it's not really persisting in the hive the way a culture does. It's an interesting point with bees, and the individual bee is super, super smart. But for passing on what it has learned about that kind of thing to other bees, its bandwidth to communicate that is really, really limited inside the hive, which I think is actually interesting in terms of how we compare the intelligence of a collective of bees versus the intelligence of an individual bee. David?
Yeah, you very quickly mentioned macaques, but an obvious thing is human babies. What age can they actually do this?
I don't know. Good question. What age can a human baby solve an abstract concept learning test?
They can't do it when they're like 1 month old. I mean, this is Piaget and all that kind of stuff.
Yeah.
Babies are dumb as logs.
They have persistent experiments, right?
And they very, very slowly develop the ability to tell their toes from their fingers. I mean, they're dumb. At what age can they actually solve these tasks?
I don't know. Object permanence kicks in about 9 months in a baby. I don't know about something like this where you're trying to test them with same and different. Because the only thing you can rely on is kind of the baby like, oh, expression, like, oh, which works in object permanence, but might be harder to apply with this. It's almost certainly in the literature, but I just don't know it, David. Sorry, mate.
Are some bees more intelligent than others?
That's interesting. So there's been some work comparing the males, queens, and workers. Intelligence is a hard word to apply here, but in terms of learning of simple associations, the workers seem to be more proficient than the drones and more proficient than the queen. What's interesting is to look at this cross-species, I think, in terms of different species of bee. Because one thing I want to emphasize as well is that I do all my work with honeybees. It's not clear to me that the bee is an insect genius. I think there could be many other insects that might have a capacity for this type of task. We just simply haven't looked. If we look at other bee species, certainly we can get, and we're doing a little bit of that work now, we can get very rapid learning and a fantastic memory in a range of bee species. But we haven't put a whole range of species through this type of task, just because the training is so arduous and because no one gives you money to do that. Yeah. Yes?
How well can they smell the sugar water?
So the good thing about sugar water, and we're pretty careful with this, sugar water itself has quite low vapor pressure. So only if they are just hovering above it would they be able to smell it. It's the same with quinine. But we always put in tests where we either have nothing at all in terms of those solutions, or we just put water into everything, into our learning tests, just in case there's a possibility that they can see. In my lab, what we do is we put the sugar water or the quinine in hidden pots that they have to reach a proboscis through to be able to taste. And that helps to remove any possibility they could see it or smell it.
So in the case when they were hovering over the paintings and choosing, is it possible they were smelling them? Very similar to the way I guess they do with flowers?
Yes, that's right. But that's also why in the test we put sugar water on everything.
Gotcha.
But certainly it is not impossible that they could have been using that cue in the training. Yeah. Yep.
You said they had like a genome of about 950,000? 900, yeah, close to 1 million? So how much variation is there across a hive? And like, do bees are hives very homogeneous in their genotypes?
So I said that they've got about 960,000 neurons. The genome has about 3,000 genes. The honeybee is genotypically quite heterogeneous. So honeybees are one of these eusocial species where the queen multiply mates. So most of the female workers in the hive are related to each other as half-sisters. Same mother, different fathers. There's all sorts of evolutionary theories as to why that's advantageous. It basically comes down to the idea that you can get a better spread of caste capabilities in the hive if you do that. Any other questions? Okay. So this problem. How on earth do you get abstraction out of such a simple brain that is only using a mushroom body as a classifier to do it? Bugged me for a long time. I took that problem to James Marshall's group, and between us, we kind of came up with this model of the mushroom body with a biologically plausible feedback neuron, the protocerebral tract. And then we used a computational modeling approach to probe the capacities of that structure to solve this task. And I'm not going to dig massively into the details of it, but we created a virtual version of the task, and we used that mushroom body model as, if you like, the brain of a virtual agent solving the match to sample and the not match to sample task in a virtual environment. We were then able to compare, well, first of all, we could get it to learn the task, and we could compare our model outputs with the actual biological data. The biological data are the pastel colors, the model outputs are the hard colors, and we could get our model to actually transfer the learned rule of same and different into a novel stimulus situation. Now, this is the paper we came up with, which is actually worth a read because it gives you the full detail of the model, explains in detail what we think is going on. And the beautiful part of a computational modeling approach is we could do what we couldn't do in the biology, which is selectively lesion every single part of our modeled mushroom body to ask which part is contributing to the capacity to do this. What we were able to work out is that what the model was learning, open question whether it's what bees are learning, what the model was learning was if we trained it on same, the model was going to what it had just seen. If we trained it on difference, the model was avoiding what it had just seen. Now, in many ways, that's the same as the statement same and different, go to what you've just seen, avoid what you've just seen. It's also not the same as the pure abstract concept of same and different. And for me, what's nice about that is it illustrates something that plagues comparative cognition, these superficial similarities. We come up with a task that we think is demonstrating something. We then realize there's potentially multiple computational solutions to that one task. And because a bee is solving this task, I said it, doesn't mean it's processing abstract concepts same and different in the same way that I am, or a macaque is, or a pigeon is, or a one-year-old child is. But certainly we were able to solve this problem of how on earth could you get something that looks like abstraction out of a mushroom body. And it speaks to the capacity of the mushroom body as this powerful classifier of multimodal information. Basically, it allows the bees to learn any cue that is going to more reliably help them to locate something that gives a reward or avoid a punishment. Now, the other thing that's come up, yes, mate, go.
So are you saying that the inference in the model is biologically plausible, or both inference and training? So there is...
So the inference in the model actually is biologically plausible. Because one of the inferences from this, go to what you've just seen, is it's time dependent. So the inference there is that if we temporally separate the stimulus at the start of the maze from the choice point, a bee's performance should fail because they're having to carry it in working memory, go to what I've just seen. Whereas if you learned a pure concept of same, I've seen this, I've got to go to the same, that should be able to be held independent of working memory. And what's nice about that is the experiment has been done. The bee's performance in this task is temporally dependent. If you stretch the maze, the bee's performance fails. So the model was able to make an inference that actually ties back to the biology of the organism in terms of the errors that they make. There's a question there then, Erica.
Did you use the output of the model to come up with new tests for the bees? So you said that the model was not actually learning same and different. It was learning avoid what I just saw.
Yep.
Did you add stimuli back to the tests for the bees to see if you could differentiate the actual computation?
We did a bit of that. We did a bit of work extending the maze and stretching it, and we did show that it was sensitive to the temporal dimension, basically what they were holding in their working memory, which is different to the macaques. We haven't probed it more than that. But that was a way that we were able to take the inference from the model back into the biology, but that's all that we've pushed it so far. Erica?
I think I just don't understand how the concept of same isn't also temporally dependent. So if because you were saying
Yeah.
that go to what you just saw will break if you stretch the time long enough. But if you see something and then you have to find the same thing or remember the same thing, it shouldn't be. But like to me, you still have to hold that in working memory. Like I don't understand how same wouldn't be broken because there has to be some memory component. Otherwise, how do you know what the ground is?
Yeah. The human experience shows that we can break that. So I could say to you, hey, Erica, here's a blue ball. They'll ask you about this tomorrow. And then I come in tomorrow and say, hey, Erica, here's a red cube. Is that the same as what I showed you yesterday? And you'd go, no. And when they do this in macaques, they've shown that they can basically stretch it over 24 hours. And they've got it. So it's
But it's still encoded, and there is a
oh, yeah.
dimension. It's just not working memory.
Yeah, it's not working memory. Working memory is like what the engram I've got currently running right now. Yeah. So it's still encoded, it's still memory dependent, but it accesses different types of memory mechanism rather than just what is currently active.
Yeah, thanks.
Yeah. Yes, mate.
I'm assuming that you could test for this by showing a stimulus and then showing a mask that it has to pass through before making the choice.
So we're doing that work right now. Yeah, we're doing that work right now. So I haven't got an answer for you, but I've actually got a student who's asking that question right now. And we're doing a couple of versions of that. So we're both putting in a mask, but we're also throwing in a different task. So you've got to hold this thing in working memory and solve a really, really simple spatial detour and then come back. So we're actually trying to interfere with the working memory. Yeah. Janie will be very pleased you asked that question. Good. Yeah. Yes, mate.
So for those of us working on computational models of different kinds of memory, could you maybe elucidate in your model how you dissociate between working and long-term memory or this memory that you're talking about, and how there is a combination of not just long-term memory, but also the fact that it needs to be matched with something? So it would be great to know the task memory versus the item memory. I would love to hear.
It would be great, and I can't give you the details on that. The work we're doing right now is we've realized that the concept of working memory is quite well established in comparative psychology and is talked about a lot in primatology. The concept of working memory has not been explored in invertebrates. Yeah. And the work we're doing right now is to ask, okay, to what extent does the concept of working memory translate at all to an invertebrate system?
What about long-term memory?
Long-term memory we're safe on. So long-term memory, I'll just jump back through my slides, make me feel a little bit sick for a second. There we go, where's the mushroom body? Oh, we're going all the way back here. Okay, too far. Long-term memory, thanks to the fly people, and just thank you, fly community, because we steal the entirety of fly neurobiologists, import it into the bee, and pretend that we made it all up. Long-term memory in a bee are structural changes in synapses at the output to the mushroom body. And you can see them, you can count them, you can know the temporal dynamics by which they form and how stable they are. And that's our best model of it. We change the output of the mushroom body, which changes how stimuli are, the valence of stimuli attached to things. So that's long-term memory. The fly communities have also worked out beautiful neurochemical phases of short-term memory, anesthesia-resistant memory, mid-term memory, and long-term memory, all of which are various changes in synapses at various parts of the brain, and or maintained cyclic AMP currents inside cells that are part of the mechanism.
And do they have sequence memory or just item memory in these experiments?
Sequence memory or item?
Like ABCD, or just item A?
Okay. That's another question that we're actively looking at in the lab, is the capacity of bees to handle sequences. And again, you'd think it had been done with invertebrates. It hasn't been done. But I've got a couple of students looking at that right now with some really, we think it's one of the cognitive limitations in the insect brain. They're not good at that. But I've got a student working on working memory, and we started off thinking like, all we talk about in insects is short-term memory. Short-term memory is not the same as working memory. And the concept of working memory has not been richly explored in insects, but we're looking at it right now. The sequence thing is fascinating in that what type of properties of a stimulus enduring over time are they actually able to hold on to? And we've just started that work. They can do something, but they probably can't do too much. Any other questions on that? Erica, I'm doing fine.
You're doing great.
Good. Okay. Yes, mate.
Maybe a naive question, but this kind of the dance patterns should show that there is some working memory because they can tell somebody else where the nectar is.
So you're absolutely right. The bee is retaining information on the location of a food source when it returns to a hive. And that's a form of working memory. We've actually got a better sense of exactly what the engram of that is, probably than anything else. There is the navigational regions of the insect brain, and Barbara Webb from Edinburgh has recently done beautiful work on this. There's a structure called the central complex at the heart of the insect brain. It reverberates and is able to maintain basically a flight vector across time. And that, in a way, that's a very specific form of working memory, which is spatial, which is probably they're then using that to replay the dance inside the hive. But that's very tightly bound to that one behavior. And again, the question is whether there's a generalizable capacity for working memory, which is more general and less tightly bound to that. But Barbara Webb has got a paper recently that's really elucidating those mechanisms, and it's really, really good. Yes, mate.
Is it the case that if you scale up the size of your model, that the performance improves, or how does the performance change?
So Alex did do a little bit of that work in terms of playing with the relative size of the feedback neuron and the size of the mushroom body. It's actually quite complicated in terms of what happens there. Either they get better at learning, if I remember this rightly, James, either they get better at learning the elemental sequences of like yellow and blue, or they get better at learning the relational differences of same. But there is a relationship in relative scale of the components of the mushroom body. I've always wanted to do some work where we actually add layers to the mushroom body, but we haven't done it yet. But we could, with this group, we could do it, but we haven't done it yet.
So regarding that, I don't know if this question exactly makes sense, but in the fly, like how deep is that module? Like, what's the longest length of a chain of interactions between neurons that you have?
Fly connectome people, help me out here. No fly connectome people. Good, I can make shit up. I don't know. There's a really good, the fly connectome papers just came out. The overview of it is a paper called Lou, which I read last week. I'm jet lagged, I can't remember. But it does talk about the path lengths through the totality of the fly brain from sensory input to motor output. The safest thing I can do, mate, is I can send you that paper rather than say something that shows my ignorance. The mushroom body to pre-motor output is relatively shallow. And think of it as a shallow brain rather than a deep brain.
But shallow, we're talking like 330, 300? Like what's the, I don't know what, I really...
Closer to 3 than 30.
Okay.
Yeah. Can I be as vague as that? And then I'll send you the papers in the break, and then you can tell me? Yeah. Any other questions? I'm going to fly through this next section. One thing that has struck us repeatedly is when we do these bee experiments, the bees learn these kinds of tasks, these kind of rule or relational learning tasks quickly compared to other organisms. So here we've got how well they do. Bees get things right about 75% of the time in this kind of context-dependent learning task, and they learn it in about 30 trials or less, as compared to various vertebrates, which don't necessarily do a huge lot better, but take hundreds to thousands of trials to learn it. Now, the problem is we are comparing chalk and cheese here across multiple dimensions. This is data harvested from the literature. All the bee stuff we do tends to be in free flight. Most of the vertebrate stuff tends to be done in a Skinner box type paradigm, as well as their different species. We really can't compare this and say bees learn faster. We tried to do an explicit comparison where we compared bees, bumblebees and honeybees with hummingbirds because we can all train them in the wild. They're all nectivorous. They're all highly rewarded by sugar. They're all highly visual and highly respond to color. We set up what is logically the same task, which is a condition-dependent discrimination, which in the bees, if it saw white, for example, blue is rewarded. If it sees black, yellow is rewarded. that kind of a task, obviously counterbalanced and all that kind of stuff. This is what the arena looked like for a honeybee, honeybee here, bumblebee here, hummingbird here. Different shaped arena because you can't get a bird to fly into a box. Logically the same task. Birds don't like black and white. They prefer orange and yellow, so we use different colors. Each time basically trying to give the organism its best chance to learn, but logically the same task. Qualitatively, we got the same result. Our bees were learning fast and faster than the much bigger-brained hummingbirds. You can't do a comparison with 3 species. We tried pushing out the species number. God, this was hard to do. Trying to get multiple labs to work across a similar sort of paradigm has been really, really hard. We have got data sets now for these species. It's all logically the same task. In every case, we're applying it in whichever way works for your species to give it the best chance to learn this. Let's not make a quantitative comparison, but a qualitative comparison does seem to tell this story, that organisms with smaller brains sort of learning faster than organisms with bigger brains. I'd love to talk to this group about why that is. One hypothesis we're sort of exploring with a little bit of modeling is that if we were looking at this as artificial neural networks, then we'd be saying that either adding hidden layers tends to slow down how long a network takes to learn. Adding noise in conjunction with adding hidden layers tends to slow down how long it takes a network to learn. So that the kind of thing we're working with here is there some sort of trade-off of more modules or more layers in your brain? Does that actually mean that you're trading off flexibility in what you can do with information over longer acquisition time? Yes, mate.
So hummingbirds and many birds have hippocampus, but honeybees obviously have the mushroom body. Is there an architectural difference and not just the size difference? Is there a comparison that you draw between the way that hippocampus would encapsulate certain information or learn sequences versus mushroom body?
Yeah, I mean, that absolutely. There are entire memory systems that are present in a bird that are not present in a bee. The hippocampus is almost like this multidimensional reference space for relating memory to memory, which a bee just does not have. So I think it's not just a matter of layers, it's about modules. I would love to tell a story that hippocampus gives you amazing flexibility for what you can do with the information that you've stored, that a bee just does not have. But the potential benefit there is a bee may be less flexible in what it's learned, but can acquire the information faster. And if you're a really short-lived species like a bee, short-lived individual like a bee, maybe the priority is learn fast and forgo the learning flexibility.
Seems like the trade-off between flexibility and speed is across different neural architectures, whether artificial or natural.
I would love to be able to tell that story, but whether our data support that, I don't know yet. But I mean, this is where some really creative modeling could really help me out, in that if we looked at modeling hypothetical brain structures, would it hold up? That we see this trade-off between flexibility and speed. And that could be a really nice story to tell in terms of cognitive evolution, if that holds up. What's holding us back is getting this type of data. It's like blood from a stone. It is so hard doing. Yes, David.
Yeah, completely independent of the actual processing, the number of layers. There are many simple models of memories, neural Hopfield nets, dense Hopfield nets, whatnot, where there's a scaling relationship between the number of memories you can store, as well as the time it takes to jump from one memory to the other and so on, versus the number of neurons. So screw all the stuff about architecture and number of layers. Well, I'm being facetious. But independent of all of that, is there a relationship simply with the size of the memory storage parts of the associated brains?
Possibly, and again, it's something that I'd need to explore, but both in terms of modeling and in terms of the biology. So, do you know, that's such a good question, mate, and we don't. We run into problems with how long we can work with one individual in terms of how many different things you could try to train it. But even dog people have run into this problem in terms of what's the memory capacity of a dog? With a dog, you just keep going. until the dog dies and it can keep learning new things. We have not probed the limits to memory in any insect that I know of. There was a really, really good paper by Mike Mangan, again with Barbara Webb, that was trying to look at it was hypothetically proposing a visual memory system based on what we think we know about how ants store visual memory. And what was asked of them is, could you hypothesize the limit to how many views an ant could store? And they tried to. They did try to. And they came up with a value of about 30 or 40 individual views that might be about. James, help me out.
I'm going to say that all because, I mean, given how simple an Infomax algorithm is, and I think the Sussex group have done this.
Yeah.
Infomax-based algorithms, you just keep storing memories.
Yeah. But I know that in that paper they were struggling because there were so many assumptions in the model of what parameters reflect what, but the editors were pushing them. Give us a value. What's the information storage capacity of this ant? And they were like, well, if the model's right, it's this. But even with an ant, even in that paradigm, we haven't been able to do a behavioral test to ask, have we hit the memory limit? We should, and we should aim to do it, but it's tough. And it's tough because of the amount of time you've got to work with one individual. Erica, I'm going to stop, mate. Because we're going to have a coffee break in 7 minutes. And rather than launching... We started a few... Do you want me to keep going? The thing is, I've got a natural break, or I go into a whole other... Ooh. Or that happens.
I mean, it's really up to you. I feel like we had a lot of questions already.
So should we just go to question? Let's just go to question time, because the next one I'm just prattling on for ages. So let's go into question time.